Okay, that seems to be everyone for now. Hello, everyone. Happy Tuesday morning, afternoon, evening, whatever it is for you, to this week's installment of Paleoperks. And today we're going to listen to a talk by Emilia Galli from the Complutense University of Madrid in Spain, who will be talking about terrestrial biomes in paleoecology, insights from Heinrich Walter's classification. So a bit about the format of today's seminar, if you're new here or haven't been here in a while. We'll start with the welcome and announcements. That's what I'm doing right now. Then we have the talk by Emilia, which will last around 30 minutes, followed by a moderated Q&A where you can send questions. We don't have tea time today, so make sure that you really send the questions that you want to send during the Q&A. And then a bit of housekeeping before we start. So Paleoperks values the participation of all folks interested in the paleosciences. So please remember to abide by our code of conduct during today's seminar. Please also keep yourself muted for the duration of the talk out of respect to the speaker. You should not be unmuted, but just in case, please mute yourself. If you want to ask questions, you can ask them either by sending them directly in the chat, or if you want to stay anonymous, you can send them to the questions at Paleoperks host, who today is Gwyn. I'm Nina, by the way. Or if you want to ask a question yourself, you can use the raise hand function in Zoom. Also, if you've experienced any technical problems during the talk, you can send them to the questions at Paleoperks host. She can help you out. We have closed captions set to English built into Zoom, and you can use the CC button to show or hide them if that makes it easier for you to follow along. And then finally, Paleoperks only works because we get so many cool early career scientists that want to give a talk or that know people that want to give a talk. So if you know someone who could give a great Paleoperks talk, or if you want to give one yourself, we also very welcome self-nominations. Please go to the link below here that will be dropped in the chat shortly. We also really encourage we also have a weekly feedback form that you can fill out just for demographic purposes that helps us to improve Paleoperks. This is anonymous, optional, and very encouraged. And the link to this feedback form will also be dropped in the chat window. Then enough about us and more about our speaker, Emilia Galli. So Emilia did her bachelor's in biology at Complutense University of Madrid in Spain. She then shifted to paleontology for her master's and currently is a PhD candidate also at the Complutense University of Madrid in Spain. And with that, Emilia, I would like to welcome you to share your screen and start your talk. Good luck.
Thank you. Okay, can you see the presentation?
Yes, it looks great.
Okay. Okay, so thank you everyone for attending this talk. I'm Emilia Galli, and I will be talking about terrestrial biomes in paleoecology with insights from Heinrich Walter's classification. So first, I want to describe exactly what a biome is and what is that we are talking about in this talk. Biomes are ecological and evolutionary units into which we can divide the Earth according to their biological communities and physical environments. So some of the most common biomes that you might be more familiarized with are the equatorial rainforest, savanna, the subtropical desert. These are located in lower latitudes of the Earth, and then in middle and higher latitudes we have other biomes like the sclerophyllous woodland, the deciduous forest, or the tundra. So in this context, why do we want to study biomes? Why are we focusing on biomes instead of other ecological units? So biomes have the characteristic of being large spatial units. So this means they are the setting where ecological and evolutionary processes happen at larger spatial and temporal scales, and these processes are extremely important for the biota's evolution, whether we are talking vegetation or the fauna. And biomes are also susceptible to different physical processes. They can change the area they take in the planet. They can expand the area or contract the habitats. This change in the area usually responds to climatic dynamics, which can promote some degree of fragmentation in different biomes. As we saw earlier, biomes can be very different in the previous slide, so they will behave differently depending on the climate we have in each time frame. So all this fragmentation and these expansions affect the connectivity between the habitats, which one way or another will hinder or promote migrations on fauna or vegetation. They also affect the resource availability for the biota, and all of this can be summarized when we look at biomes in their change in geographic distribution and the landscape structure they offer to the biota. So biomes are interesting, summarizing what we are talking about, because they can be a kind of magnifying glass into the processes that drive biota's evolution, whether we are talking about vegetation or about fauna. So to see a general example on how this can be included in paleontology, we know that throughout the Cenozoic, land masses have changed their distribution in latitude. They have changed the connection between the land masses. They have also changed the topography, heterogeneity, how the climate affects them, and we can summarize this in this graphic we have in here. So here, I don't know if you can see here. So here you can see the age that we are managing right now in million years. And we have the record of the oxygen isotope, which allow us to have an idea of the temperature and the changes in temperature we have through the fossil record. Here we have how on the left we would have lower temperatures, which means colder climate. And here we have warmer temperatures, which means a warmer climate. And we know that these changes, these climatic changes that we have in here are correlated to the tectonic events that affect the land masses and the habitats we want to study, as well as the biotic events and the fauna or the biota we want to address. So a more clear example can be the quaternary glaciations, which are more recent and are widely studied. We know that these glaciations were not continuous. They had intervals of colder climates with an extension of the ice sheets, and then warmer climates with the retreat of these ice sheets. So this affects the resources availability we mentioned before, the landscape structure that offers the biotas to inhabit, and all of this affects fauna's migration, which is in which I'm more specialized. I work mostly with mammals. So this affects the fauna's migration, which includes or promotes different evolutionary events like speciation, extinction, or specialization, among others. So we are going to look at specific examples where they use biomes to look into paleoecological hypotheses or evolutionary processes. The first one focuses on rodent assemblages from the Iberian Pleistocene. The study was performed on the Iberian Peninsula, which is in Europe in here. And here the authors work with rodents from the last 5 million years, and they looked at the biospecialization of these assemblages. So what they did was look if rodents were present in one biome or one or two biomes, or they were present in more biomes. This means that rodents could be more generalist or more specialist. So they were able to link the biome specialization of rodents along these 5 million years with climatic changes during the Neogen and Quaternary boundary, and specifically they correlated these changes to Quaternary glaciations. So addressing the biomes, they were also able to address the changes in the percentage of rodent species that they observed in these 5 million years within the different assemblages. Okay, so now we are going to look at other examples that address biomes more directly. This other group of authors looked at the area each biome had, the age of origin of the biome, and the climatic stability. And they did this for a global scope by also looking at different land masses. And similar to before, they looked at the changes in geographical patterns of biomes across the last 5 million years. So doing this, they were able to test hypotheses to explain the high diversification we see today in the tropics for amphibians, birds, and mammals. In another study, the same group of authors address the historical fragmentation we see in the biomes. So they did this again for the last 5 million years, and they accounted for the number of fragments of each biome and the size of each patch. And they look at how this varied across the last 5 million years. So this way they were able to link mammals' biome specialization to historical fragmentation, as well as testing macroevolutionary hypotheses to extend the pattern that we see nowadays. So those were some examples to illustrate how biomes can be included in paleoecology and how they can be really relevant to address different hypotheses. However, we have a wide variety of studies that use biomes to perform different analysis. So here we have on the left one that looked into the current distribution of turtles through biomes. Here we have another one that reconstructed the Middle Miocene biomes based on climatic models, but also on paleontological fossil records of vegetation. And here we have another study that looks into how biodiversity distribution changes throughout the biomes along the Cenozoic. So one question that we may have is how we are able to include biomes in these studies. So how can we address them, or how can we include them in a more specific way than just the name of the biome or the category of the biome? So we have different classifications to identify the biomes. Each classification has different categories. They mostly use climate to define biomes, but they also use different approaches. So here we have 6 of the most traditional and widely used categories, and most of them have a clear identification of each biome and clear boundaries for each biome. So this quantification was possible from 2 vias. The first one being the original authors defining the biomes themselves, and the other one is modern research groups being able to quantify these biomes thanks to the availability of climatic data for today and also for paleontological scenarios thanks to the inference with climatic models. So all of this is also possible with the use of geographic information systems that allow us to include all this spatial information for our studies. So here I will focus on the classification of Walter, which is one of the traditional classifications. So this classification is very relevant in terrestrial ecology. It was one of the first ones to be developed, and it was one of the first ones to address the importance of the biotas community and the vegetation physiology to define the ecosystems at larger scales. This classification has an integrative approach that is based on climatic variables. And the author uses climatic variables to define the vegetation physiognomy of each biome, the seasonality patterns we find in each biome, the water availability, as well as the temperature threshold, all of which are extremely relevant to define the biota that we find in each biome. So this allowed the author to define different coherent plant communities that we can find along the globe, and this resulted in 9 traditional biomes. However, this classification remains qualitative and thus incompatible with modern studies where precision is needed to get solid conclusions. And this is the gap that we want to acknowledge in this talk. So the objective was to quantify this classification. Some researchers can include it in their studies. And to do this, we perform a climatic characterization, which is based on the original works of the author as well as its posterior proposals. We also work with another authors that are experts on vegetation physiology and physiognomy and the different thresholds for the biota. And of course, we complemented all of this with updated criteria from the literature. So this gave us this dichotomous key we find here. We have a variety of climatic patterns, and these climatic patterns have a different biome unit assigned to each one of them. However, when we want to include this in our studies, it still seems kind of far from the data that we usually manage. So to implement this characterization into our study, we develop a function in R. To do that, we work with the PaleoPGene database, which has climatic data from now until 5 million years ago. We work first with pre-industrial data to work on the current distribution of biomes. This database has a temporal resolution of 1,000 years and a spatial resolution of half a degree cell. We perform all this quantification of the classification of Walter with a global scope. So to do this and define all the variables we saw before in the dichotomous tree, we work with temperature and precipitation data, and specifically with mean monthly values. This is important because when we want to work with climatic data, we need to look at the climatic diagrams to understand the interplay between both variables. So here we have the precipitation, here we will have the temperature data, and on the horizontal axis we have the number of months. So for each month we will have 2 values, one for temperature and one for precipitation. And this will allow us to look at the climatic patterns like the seasonality, water availability, and similar that we mentioned before. So including all of these interplays, we were able to develop the function Walter, which I will talk about now. So this is the percent distribution of biomes with a global scope. This is the distribution that we got with this function. Here we have in the legend the colors as well as the vegetation community that each biome represents. The, the function allowed us to obtain a total of 10 biomes. These are built above upon widely available data. As I mentioned before, we use only two variables, which are widely available not only for present day studies, but also for paleontological ones. It has no temporal or spatial restriction and is built to be freely accessible for many researchers. So to see more specifically each biome and understand which kind of community it entails, we are going to look them individually, very quick. So here we have the equatorial rainforest, and here you have an image of how the vegetation would look in each biome. So we have the temporal deciduous woodland, the savanna, the subtropical desert. As you see, the biomes will follow a latitudinal organization from lower to higher latitude. We also have the sclerophyllous woodland, the temperate evergreen forest, the deciduous forest, the savanna, the taiga, and finally the tundra. So once we have this distribution of biomes for the present day, we went into the past and obtained and worked with the last interglacial. This interglacial lasts from 130 to 115,000 years ago. Here we have the chronostratigraphic context of the last interglacial. We have here the age in million years, and we will find ourselves in this red frame, which is entailed in the late Pleistocene within the Quaternary. So this is the last interglacial because right now we are living in an interglacial, which is right here. And before us, we had the quaternary glaciations that we mentioned before. So here is what we want to look at. And here we have again our graphic with the isotopic record of oxygen indicating the temperatures, here the lower temperatures and here the warmer temperatures. And we can see that at the beginning, the last interglacial had a very fast warming trend, which became more slower towards the cooler trend and the posterior glaciations. So when we applied our function to the last interglacial, we found we were able to address these climatic dynamics every 1,000 years according to the resolution of the paleopgm, since we use the same database. So we can see that at the beginning of the last interglacial, we have much cooling biomes and more arid biomes. Can you see it? Okay. And then towards the end, we have warmer biomes. As we can see, the most forested ones can reach higher latitudes, and then they retreat again and steppes become more dominant. We can also see that most of the changes happen in the northern hemisphere, where we have more land emerge, whereas in the southern hemisphere, biomes remain much more stable and more subtle, and changes are more subtle. So if we look in detail into the last interglacial, here we have a schematic image to look into the details. Here is the legend again to see how the different vegetation communities change across time. At first, we have the warming trend, and we accompanied our reconstruction with paleontological data, which is very important to contrast our results and make sure our function works for past environments. So the paleontological data show that mixed forests with close canopy reach higher latitudes than we have today and than we had before at the beginning of the last interglacial. This record also showed that the boreal forest reached up to the Arctic coast, which nowadays is not the case because the tundra takes most of the coast. And we also found that humid biomes reach middle latitudes, whereas before they were more restricted to lower latitudes. When we look at the cooling trend, we see that arid biomes and cool and more cold biomes were more dominant. We see that steppes become more relevant now, and the fossil record shows a lot of taxa typical of open vegetation being more dominant in middle and higher latitudes. And they also show that woodlands were restricted to the southern regions of the northern hemisphere and in lower latitudes. We can see these changes, for example, when we go from the beginning of the last interglacial towards the warmer trend, we see that the temperate evergreen forest, which is this one, appears in Europe and is more developed in North America, and it disappears from Europe and it contracts significantly in North America. So with all of these, biomes show a great potential for paleoecological inference at larger spatial scales, whether it is for the reconstruction of past environments or to look at the biodiversity distribution, or also to address the changing patterns of both through time, even if we look at only vegetation, or also at the fauna. Biomes are also relevant to address macroevolutionary processes of both extant and extinct biota, especially those related to the biogeographical history of biomes, since we have seen that fragmentation of the biomes are very important for the evolution of the biota, the area they occupy, the climatic stability, as well as the connectivity between the habitats or the resource availability for the biota. Finally, Walter stands as a new alternative which has a great potential in paleontological studies since its integrative approach allows for a more comprehensive context in paleontological and evolutionary interpretations. And with this, I want to thank everyone for attending the talk. And now, if you want to proceed with the questions, I'll be glad.
Thank you so much for your talk, Emilia. This was super interesting. Brief reminder for the audience that you can send in your questions now. We have already received a few, so I will just start with those. So I will drop them in the chat and I will also read them out. So the first is an anonymous question, and this person asks, how far back in time can we reliably compare model biome results with proxy-based inferences of temperature and rainfall?
This is a very interesting question. Thank you. We have here two important things to consider. First, paleoclimatic models have their own limitations because we are doing inferences using paleontological data, but also simulations. So we cannot look at very detailed reconstructions. However, they are very interesting to see general trends or large-scale reconstruction, which is the most important of the talk. And I want to emphasize that this is everything for large spatial scales, mostly, which is where climatic and other abiotic factors are the ones that drive these changes. We also know that at deeper times, we have less spatial resolution. So here we are using half-degree cells, which are approximately 55 kilometer cells. When you go deeper, and you can find a lot of studies, this resolution is much greater. They usually use 100 kilometer cells or wider because we are not able to reach the same precision. And other things to consider is that along through time, we know that different variables have changed, and for example, carbon dioxide. And this is very important because vegetation's response to climate depends on the levels of carbon dioxide we find in the air. So this is a reason that we chose to work with the last interglacial. First, because it is uh, the recent past, which is more reliable, and it also has the similar levels of carbon dioxide than today's. So we know that the climatic thresholds we developed are compatible with this reconstruction. And for example, if we wanted to use it for another scenario like the last glacial maximum, we know that carbon dioxide levels were very different, and thus the vegetation response would be different. So the threshold we have are not the same, and that would need a correction, which is one of our future investigations. We want to add this carbon dioxide correction, so we are able to use this function in a more variety of scenarios.
Thank you for that very comprehensive answer. I think that definitely answers the question. I also really love the tundra background that you chose for your last slide.
Thank you.
Yeah. So the next question asks, can big data tools like the PBDB, for those who don't know, that's a paleobiology database, I think, help test how the fossil record compares with your climate reconstruction, or is its temporal resolution too low?
It is useful. We actually build this function to help researchers have an easier but also reliable way to reconstruct the past. So, for example, if we are working with PBDB and the fossil record we find there, and we want to know in which environment we are having, whether the fauna or the vegetation you are working with, and the biomes provide the potential, it's focused on the potential biomes we might find in the localities we are studying or the regions we are studying. So this is important because we can know if the environment where we are working has actually the potential to develop the vegetation that we can find in the fossil record, or another option is that we might find, for example, a fossil record of taxa from open environments where the function would show a more closed vegetation environment. So that can also look into the lag we find between the vegetation response to the climatic changes, because we know vegetation doesn't have doesn't change instantly or doesn't respond instantly to these climatic changes. So it takes them some time to change all the environment. And this is a way to address these differences. And it's also an opportunity to delve into the potential environment we can find and the potential environment that we want to, into which we want to include these paleontological data that we know.
Thank you, Emilia. There is another question that is kind of related to this, like the time it takes plants to respond. Because this question asks, can the model also simulate transient biome changes, or are they all time slice type models?
Yes, this is also another thing like to consider from the function, because right now we are delimiting the biomes with very specific thresholds, and they are a more rigid boundary. However, we know that reality is not that this way. So between two different biomes, we have transition zones, which we call ecotones. So it's kind of mixed region of characteristic of both biomes that we have now in connection. So the function is not capable yet to address these ecotones or these transition zones. However, this is also a future work because including the ecotones takes a lot of more of time and a lot of more of effort. And the first goal we had here was to make available this classification that was not an option for a lot of researchers. So this is kind of a first step towards a bigger project and of course a much longer project that will build up with all the carbon dioxide changes and these ecotones that we also need to know. However, the function also allows us to know which kind of environments we might lose. So if you have a locality that is next to a biome boundary and you have some mixed data or mixed results, the function allows you to know if you're actually if the environment you have is potentially one biome, or maybe you are looking at a transition zone. So with the function, you could address conflictive data or conflicted results that suggest different environments, which is something that usually happens in the paleontological records. And this could point towards a transition zone that we can look with our function. So we will know, okay, maybe we are in the tropical deciduous woodland, but we are near the savanna. So maybe we are looking right now at a mix of these two biomes, and that would be like a first approach to address this integrative interpretation.
Thank you again for that answer. I think I have a question that is, again, kind of similar to what you already explained. Is it asks, are there any biomes that are particularly difficult to reconstruct or model? For example, because they are very similar to another biome, or because the model and proxy results often disagree?
Yes, finally, we had some biomes that were much harder to define than other ones. For example, the tundra was very easy because all you need to have is extremely cold temperatures, and it has to be humid, but you're still able to to have this biome very easily defined with temperature. And the same happens with subtropical desert. You have extremely high temperatures. So whether you have more or less precipitation, you know that temperature it can be extremely relevant to define them. However, when you go into the not transitional biomes, but maybe intermediate biomes like the temperate evergreen forest, which is, let's see if we can go back. So here with this one was much harder because it's usually included in the deciduous forest into this one. So in other classifications or in other studies, they look like one gigantic biome, and they're actually different. They are so different that this biome appears actually in the higher altitudes in mountain ranges in tropical biomes. So this was much harder. This required us a lot of work, a lot of research, also to look at meteorological stations where we could see the climatic diagrams we mentioned before that were so important to define the biomes, these ones over here, and look at how the interplay between precipitation and temperature let us know if we were actually in the temperate evergreen forest or the deciduous forest, or in the case of the lower temperatures, if we were in this biome or we were in the equatorial rainforest. So this one, and we also look at the literature looking for native vegetation to make sure that our climatic reconstruction matches the vegetation that we find nowadays in these localities or these regions.
Thank you. That's very interesting. I think so far we have one question left,
Okay.
which I think is very interesting. They ask, how could your methods be adapted to reconstruct marine climates? And can you resolve biome boundaries of coastal ecosystems?
This one is a very interesting question. Thank you. We know we have other biome classifications for marine environments. However, they are extremely different from these ones because being in the water, the temperature interplay with physiological thresholds for the biota are they're like completely different. And here we also have in terrestrial biomes, precipitation is extremely important because they don't have the water availability. It has an uneven distribution throughout the globe. And it's one of the restrictions for the development of life. If you don't have water, you cannot have life. However, in marine biomes is completely different. So the classification would be you should build the classification on different variables, considering of course different thresholds because in the case of precipitation it wouldn't be a restriction. We should look more at sunlight availability, the temperature threshold. It would be also be much more important the saline concentration, for example, and I'm sure the oxygen concentration. So these are more out of my field of expertise, but I know they also follow a similar latitudinal gradient because they are in the same system. They are not in they are in the same planet. However, the variables affecting the biota are very different. So I don't think our function would be able to work with this kind of data. I think you should look into other proposals which are much more accurate and actually work with the relevant relevant variables for marine biomes.
Yeah, thank you. That makes sense, I think. To the audience, if you still have any questions, please type it in the chat very quickly. Otherwise, I already want to thank Emilia for the very interesting talk and for very extensive answers to all your questions. I also want to thank the audience for all their questions and their interest. And if there is no more questions, then I want to just end by sharing some information on upcoming paleoperks seminars.
Okay, thank you very much.
So yeah, thank you again, everyone, for joining us. Thank you again, Emilia, for a really cool talk. Please remind you to please fill out the feedback form again, if you would be so kind. It would really help us. Then in the coming days or weeks, we won't have any seminars. For a lot of people, there will be holidays or just a break. If you're celebrating anything, I wish you a very great time. If you're just having a break, I hope you have a very relaxing break. And in any case, I hope to see you back in January, because on January 13th, at the usual time, we'll have a talk by Alessandro Carniti from Nanjing University in China about paleoecology and sedimentology, and more details on that will follow. With that, happy holidays if you celebrate, happy New Year, and I see you in 2026, I hope.